Okay, so let's go ahead and get started. First off, I want to say thank you very much for joining us today. We're very honored and privileged to have you here today to join us for our interactive workshop on viral infection modeling using CompuCell3D. I'd like to start this session by all of us introducing ourselves. So my name is TJ Sego. I'm a postdoctoral fellow at the Biocomplexity Institute at Indiana University, which is directed by Professor James Glazier. So Professor Glazier, if you could introduce yourself.
Hello, I'm Professor Glazier, James Glazier, and I welcome you today. I'll be acting as a moderator. If you have questions, type them in the chat and we'll get you to the people who can answer those questions. Thanks very much for joining us.
Thank you, Professor Glazier. And Josh.
Hello, everyone. Thanks for being here. I am a PhD student at James Glazier's lab, and I'll be available to help people with Mac issues in the breakout room. Just send a message and then we can meet there, and I'll help you guys get CompuCell running.
Thank you, Josh. And Giuliano.
Hello, Giuliano. Basically the same thing as Josh, a PhD student with James Glazier. The only major difference is I'm here for Windows people instead of Mac people.
Thank you, Giuliano. And last, Gilberto.
He was going to be late today.
Okay,
remember?
Great. Oh, that's right. Okay, well, Gilberto will be joining us later in the workshop. He'll be joining us from Brazil. As last mentioned, before we proceed, we've got the project tool available in Nanohub. That's our recommended implementation to get through the workshop today. We'll have a little time before we get into that. So we're going to start today with a kind of flyby of some of the relevant biology that we've been particularly interested in with modeling viral infection, as well as some of the basic notions of computational modeling and abstraction of those biological concepts. We'll then do a brief overview of the model that the simulation framework consists of, implements at a very high level, as well as survey some key results that we featured in our preprint that we've recently submitted. You can find a link to access that preprint in the chat at the very top. After that, we'll do a brief question and answer session, and then we'll take a break so that everyone can rehydrate, recaffeinate, whatnot. And then after that, then we'll dive into playing with the framework. For this session, we're going to stick with basic experiments with model parameters. So no real coding for this session, but instead we're going to interrogate the model and some particular parameters of interest to see what we observed due to those changes. So the way that we're going to do this, to keep this flowing smoothly, we've disabled screen sharing and microphones for all attendees. So please, for any questions or comments, feedback, any issues, please enter those into the Zoom chat. As Professor Glazier mentioned, he'll be moderating if there are any issues with software, installations, or otherwise. We'll move people into break rooms. Josh and Giuliano, as they said, they're here to support. So please enter all of that into the chat. And with that being said, let's proceed. And for as excited as we are to be here and to host this event and have all of you here, it's then sort of bittersweet to start on such a dreary note. But of course, we're most of us, myself included, are calling in from quarantine, from our own little personal prisons that are our homes. And what we would like to do is really understand what about this virus makes it so difficult, makes it so difficult to predict. We know that, for example, patients who have been infected are infectious before they're symptomatic, makes it particularly difficult to track and to contain outbreaks. We know that the patient outcomes are highly variable, both from patient to patient, as well as location geographically. It's also particularly difficult because the causes of death vary over a wide range of systems throughout the body, complications with the cardiovascular system, so on and so forth. And this is also particularly difficult for those of us who work in modeling, because coming into this as early as we are, data still is very sparse. It's very sparse, it's not well aggregated or organized. It grows by the day, and so it's very difficult to keep up with. But we sort of come at this with the expectation that the data will eventually be there, and we develop our models and our simulation frameworks to accommodate that and to quickly adapt to data as it becomes available. So as modelers, we have a lot of questions that motivate us when we think of how we might be able to contribute to solving this problem of SARS-CoV-2 infection. And we're really curious about why some people get sick and why other people don't. Why some people don't even show symptoms and are what are called, often called silent carriers. We'd like to understand why COVID is so much different than something like influenza, something that's better understood, as well as why there's so many delayed adverse responses in individuals and complications associated with pneumonia and cytokine storm. We'd also like to know what are the particular mechanisms associated with age-related effects and why the elderly seem to be so much more adversely affected. And then in application, biomedical applications, we'd also like to know, is there something that we can build? There's something that we can simulate to assist in, say, performing better triage, or assisting in drug discovery and drug therapy development by optimizing antiviral or anti-inflammatory therapies. And so we do this for the sort of modeling that we do by asking the basic question of, can we predict systems level responses due to perturbations at the molecular level? And so we come at this by first starting with aggregating the sort of biological components that are relevant to our problem of interest, in this case, SARS-CoV-2 infection. And in our case, this is a particularly difficult problem because the most relevant biological components tend to span a lot of scales over the entire body, all the way down to the subcellular molecular level. So in our case, we certainly know that we're interested in the target epithelial tissue of infection and all of the complexity associated with that, like its extracellular environment, the mechanisms of viral replication, like internalization and viral release. There's a lot of complexity associated with the various types of immune cells that we find in a particular epithelial tissue and how they're recruited to that local tissue, and what all sorts of things those various immune cell types do. There's a lot of complexity concerning the signaling involved with getting them into a local site of infection and how they communicate amongst each other and with infected cells. And then, of course, there's lots of tissue damage that occurs due to infection and immune response, and hopefully in a positive outcome, tissue recovery. Now, at the higher level, sort of the systems level, there's a systemic immune response that has a lot of complexity to do with signaling with the immune system from a local site of infection to more of the systems level immune response that induces things like cell proliferation, systems level migration from proliferation sites to sites of infection. And this involves a lot of complexity concerning whole body transport through blood vessels and the lymphatic system, There's also some complexity to do with how the virus gets into the body in the first place, with something like air. And then, of course, there are issues associated with complications that arise due to secondary target organs like the heart and the kidneys that bring about all sorts of complications that cause fatal outcomes. Now, the immune response at the systems level has an immense amount of complexity all on its own. And we've constructed this schematic here that's sort of an aggregate of schematics that we've found for a couple of different types of viral agents to sort of give it more of a general sense of what a general immune response would look like to something like a viral infection. Now, there's an early innate immune response where immune cell scavenging and phagocytosis of virus begins almost immediately after infection in the first couple of days. There's then interferon signaling that induces viral resistance by infected cells. Systemic cytokine signaling then starts usually around 2 days. This gets relayed and amplified by immune cells to kick in the mechanisms to start the adaptive immune response. Natural killer cells are recruited to a local site of infection to do early killing in the innate immune response. Dendritic cells will then leave a site of infection and travel to a lymph node and present antigen. And this induces the return of CD8+ and other T cells to a site of infection for pathogen-specific cytotoxic killing. This also induces B cell proliferation and the production of antibodies in the adaptive immune response, which eventually leads to immunity and long-term resistance to an infection. Now, when we look inside individual cells, or when we look at a particular population of cells and measure the amount of virus that we find that after infection, there's an initial eclipse phase. You can see this both for data on influenza and for SARS-CoV-2. There's this eclipse phase after initial infection where we really don't find much virus, after which there's this sort of rapid exponential growth. This is due to exponential amplification by genomic replication. And then afterwards, we find this sort of saturation due to the effects of the immune response beginning to deal with the infection. And then, again, in a positive outcome, we'll find that the amount of virus in a particular population will then rapidly decline towards viral clearance. Now, damage of tissue by cell death begins typically after the first couple of days, and depending on the severity of infection, will get much worse throughout the progression of events of infection. And cell death occurs due to a lot of mechanisms. So the virus itself can induce apoptosis due to its own replication. There is the induction of cell apoptosis due to antiviral signaling, like the type 1 interferon signaling from infected cells. And then, of course, immune cells induce cell death due to cytotoxic killing, both due to surface interactions, contact killing. There's then what's called a bystander effect, in which cells that neighbor an apoptotic cell will also become apoptotic. And then, of course, there's the secretion of reactive oxygen species that will kill infected and uninfected cells alike. Then again, in the case of a positive outcome from infection, typically cell proliferation from stem cell-like local cells will begin somewhere around 10 days after initial infection. And so we take these ideas and then we think to build a model, we have to take these relevant biological components and then we abstract them through some sort of conceptualization into mathematical objects. And so for our case, our 4 main general objects of interest are epithelial target cells, replication of the virus, internalization, and other associated events. Then there are immune cells, and then systemic immune system recruitment and response. So in the case of epithelial cells, when we look at something like a lung epithelium, when we look close enough at something towards a cellular level, we find that epithelia are about two-dimensional, with various forms of stratification. We find that the epithelia will vary by cell type and location, as well as geometry, depending on where we look, and that individual cells can vary in their susceptibility to viral infection. Now, in our case, we begin by starting with a two-dimensional sheet of epithelial cells, and this sheet we'll consider as being homogeneous. Every cell has the same susceptibility and is the same sort of epithelial cell type. For viral replication, depending on the virus, replication is very complicated with lots of steps. But one thing particularly of interest is that, say in the case of SARS-CoV-2, there's the mechanism of RNA replication that leads to exponential amplification of the number of virus inside of the cell. So we know that the virus will somehow get into the cell. We know that due to genomic replication, exponential amplification will occur, and then afterwards, virus will be released from the cell back into the extracellular environment. And so we do a representation of this using a system of ordinary differential equations. The set is shown here. In particular, you'll notice that we model nonlinear exponential amplification in the replication stage for genomic replication. And then there's this figure here shown on the right. This is an example of the system being simulated in a particular cell. We model an eclipse phase after infection before any virus is released back into the extracellular environment. Then you'll notice sometime after that there's exponential growth and then up to some saturation. For local immune response, there are a lot of immune cells that do a lot of things at a local site of infection. These various immune cells do lots of different things, like phagocytosis. There's a lot of interferon signaling between infected and uninfected cells. Immune cells do cytotoxic killing by various mechanisms, as discussed before. There's a lot of cell and signaling activity over time that's very complicated. There's antigen presentation and transport to the lymph nodes for recruitment and activation of the adaptive immune response. Cytokine signals secreted in the local site are then relayed and amplified by immune cells. There's a lot of complexity with the pattern of infiltration into a local site of infection by immune cells. You know, in the case of both SARS-CoV-1 and SARS-CoV-2, that there is the infection of both T cells and macrophages, as well as their depletion over time. We know that in general, when moving through the progression of infection, that we'll transition from an initial innate immune response to more of an adaptive immune response due to all of the antigen presentation and immune signaling. Know that non-specific antibodies may play a role in containment of infection. And then, of course, there's specific antibodies that are produced and released later in the adaptive immune response. So to begin by describing all of this complexity, we start by saying that we have an immune cell type and one cytokine field. And we say that on top of our two-dimensional epithelial sheet, we'll introduce our generic immune cell type on top of that sheet. And the immune cells will stay frozen in their epithelial sheet, and the epithelial cells will stay frozen in their epithelial sheet on their level, and then on top the immune cells will be modal, and they'll go about doing all of their immune cell killing, and so on and so forth. Concerning the systemic systems level immune response, we know that Immune signaling at the systems level consists of a complex mixture of both pro-inflammatory and anti-inflammatory signals that regulate response throughout the events and progression of infection. You know that there's local signaling in a site of infection that recruits circulating immune cells as they circulate by. You know that this then leads to long-range signaling to the systemic immune response, and that this upregulates immune cell production. We know that there's the migration of antigen-presenting cells from a site of infection to lymph nodes, and this leads to the production of antigen-specific T cells, as well as B cells and pathogen-specific antibodies. Now, we represent this with a compartmental model with an ordinary differential equation that represents long-range signaling from the local tissue. Now, the long-range signaling determines either the net pro or anti-inflammatory state of the immune response. We then say that there is some distant lymph node that then will supply immune cells to our local site of infection based on the pro or anti-inflammatory state of the system. And then to represent something like the distance that's traveled by transport through the lymphatic system and so on and so forth, we then say that when we represent this lymph node and systemic recruitment, that there should be some sort of delay between the local signaling and the corresponding arrival and recruitment of immune cells to the tissue patch. Lastly, concerning tissue damage and recovery, viral production, for example, has both intrinsic and extrinsic cell death pathways. There's also all of the mentioned mechanisms about immune cell killing of infected cells. We know that there's also cytokine-induced damage in epithelial cells. And then later, the corresponding recovery of a tissue patch after damage has occurred and the virus has been cleared. So for our case, for the time being, because of the amount of time that we simulate, we've not really explored much in modeling the tissue recovery for now, but we certainly represent cell death. This is a sample of our simulation results. This is an area of damaged tissue with dead cells. And we include cell death mechanisms associated with both production of virus inside of individual cells, as well as representations of immune cell killing of epithelial cells by contact, bystander effect, and oxidative killing. So now when we do this, we consider all of these biological concepts and abstracting them, we find that when we put them together, that these modeling concepts scan 3 different scales. So we have a cellular tissue patch of epithelium with immune cells on top doing what they do. And there's a global signal that's sent due to the aggregate behavior of the entire cell aggregate. That global signal is transmitted to our compartmental model of the lymph node that leads to recruitment of immune cells to the tissue patch. Meanwhile, when we look in an individual cell, as shown here, in any individual cell, we find that each of them has to have a representation of viral replication according to the state of the particular cell. When virus internalized, how much virus was internalized, so on and so forth. So we do this in CompuCell3D. CompuCell3D is a virtual tissue modeling and simulation environment that we develop here in Bloomington at the Biocomplexity Institute. This simulation framework is open source. We spend a lot of time providing training, both in using the methods that we include in CompuCell, as well as training for developing CompuCell for specific applications. One of our ultimate goals is to allow for clinical and industrial researchers to develop models on their own without requiring any sort of expertise in doing computational modeling itself. So we strive very hard to include a lot of accessibility and reduce the barrier of entry to doing model specification in our software package. And you can find just the project website here at CompuCell.org. Now, we do this by building computer simulations to explain how interacting mechanisms lead to specific outcomes. Specifically, for problems that involve multi-scale interactions, like viral infection, the systemic response to local infection, to subcellular events, and particularly when trying to extrapolate information from individual molecular signals is insufficient to explain the mechanisms for something that we're interested in. We do this to try to understand disease mechanisms, hopefully to identify critical pathways for treating a particular disease. We also try to do this to identify drug targets, specifically for currently untreatable conditions. We also have a lot of interest in personalizing existing therapies in something like personalized medicine and digital twins, as well as identifying toxicity mechanisms in various treatments. Here's a very brief list of some applications that have been developed either by current or previous members of the Biocomplexity Institute, as well as collaborators like IU School of Optometry, and Environmental Protection Agency. This has been work done in polycystic kidney disease, diabetic retinopathy, liver toxicity. One thing that you don't see in this list currently is modeling viral infection. And that's what we've started here. So we've developed this modeling framework, or this model set of modules, and this simulation framework to begin doing modeling of viral infection, multi-scale modeling of viral infection using CompuCell3D. So we're going to go through very briefly the model, the various model components, not so much the mathematical representations, but the interactions of the representations of the various biological objects. And then after that, we'll go through some very brief results, and then we'll do a brief Q&A. So our goal was to start very modestly by first looking at the spread of infection in a small patch of epithelial tissue. So we modeled primary infection, assuming that in our simulations that there are no pre-existing adaptive immune response from previous exposure to our virus in simulation. And so there are no specific antibodies that are targeted to our virus, and then no memory T cells or B cells. We also assume in our simulations that we're modeling something like acute infection. And so we simulate a shorter time frame where either the immune system at the end of simulation clears the virus spreads over the entire tissue patch in our simulations, or something in between those two outcomes. So the model consists of 2 cell classes, 3 diffusive fields, and our compartmental model representation of a lymph node. So we say that we have 1 epithelial cell type, and that our epithelium consists of a set of those epithelial cells arranged in space. And then on top of that, we have our generic immune cell, and our immune cells consist of a set of those immune cell types that go about all of their activities on top of our epithelial sheet. We then also include 3 diffusive fields that are solved with partial differential equations. One modeling the transport of extracellular virus throughout the tissue patch. We also have the secretion and diffusive transport of our cytokine field used for local and global immune cell signaling. And then we have an oxidative agent that's secreted by immune cells when activated that then kill epithelial cells. And then, as discussed before, we then have our compartmental model of a lymph node that goes about regulating the population of immune cells in our tissue patch, and it receives signals from the cytokine transport module, where cytokine secretion occurs by infected cells and immune cells that then leads to immune cells being introduced into the simulation patch according to a delay. So with our epithelial cell class, we have 4 epithelial cell types. Now, these are not biological cell types, but rather we use cell types to describe the various states of infection of a particular epithelial cell. So we have 4 stages: cells that are uninfected, epithelial cells that have been infected but are not releasing virus into the extracellular space. Then afterwards, there are virus-releasing epithelial cells that are releasing virus, and then the terminal state of being dead. And now we have various mechanisms describing how the epithelial and immune cells interact, and there's a list running here from left to right. You can see that the interactions correspond to these various stages of the epithelial infected cell. So all cell types can internalize virus, and so we allow for superinfection. Of course, infected and virus-releasing cells are the only ones that can experience replication of intracellular virus, because they're the only ones with virus inside. Our virus-releasing cells are the only cell type that can release virus, whereas both infected and virus-releasing cells will release cytokines into the extracellular environment. Virus-releasing cell types can die from virus due to cytotoxic effects. And then our immune cells can kill all 3 epithelial cell types, uninfected, infected, and virus-releasing, both by secretion of our oxidative agent and by a bystander effect, which is the case when an epithelial cell is killed on contact, in which case neighboring cells then have a probability of also dying along with the first cell due to our bystander effect. For our immune cell types, again, our immune cells are the only cells that move, so they exhibit motility along the top of our epithelial sheet. Now, for our one immune cell type, we have 2 different types of immune cells. Immune cells are introduced into a simulation on recruitment as being in an inactive state, in which case they can do some behaviors like killing epithelial cells on contact. Now, in response to being exposed to cytokine by signaling from infected cells, the inactive immune cells can then become activated, and they become an activated immune cell type, in which case they then will chemotax along gradients of the cytokine to find virus. They also will kill infected cells on contact, but activated cells will also release cytokine to represent relaying an immune signal. And then in sufficient exposure to cytokine, an activated immune cell will also secrete the oxidative agent, which, as mentioned, does cytotoxic killing of uninfected, infected, and virus-releasing cells. Now, to represent the basic processes of viral replication, we have 3 stages. As discussed before, there's the event of virus getting into a cell, and this is modeled as being proportional to the number of receptors available on a cell's surface, as well as the amount of extracellular virus that a particular cell is exposed to. We then represent viral replication. This is represented as occurring over 4 basic stages. There's a first stage of unpacking after a virus is internalized. There's then the exponential amplification phase of genomic replication. And then after replication, there are then the stages of protein synthesis and assembly and packing. And assembly and packing, these are the stages that affect virally induced apoptosis, as well as the last stage of basic viral replication, which is release of the replicated virus back into the extracellular environment. Now, immune recruitment is again represented with an ordinary differential equation. We tend to refer to this equation as just a state variable S. And what we say is that when our ordinary differential equation S is greater than 0, the immune system is then in a net pro-inflammatory state, in which case the probability of adding an immune cell to our local tissue patch is greater than 0. So the higher and non-negative S is, the more likely we are to find immune cells being added to the patch. Whereas when our state variable S is less than 0, the immune system is in a net anti-inflammatory state, in which case we are more likely to find immune cells being removed from our local tissue patch. Now, with that convention in mind, we then take two particular pieces of information about our spatial domain and we map that onto affecting our state variable S. And we say that when the total cytokine level in the entire spatial domain is great, then the immune signal S will increase. Whereas when we add more immune cells into the domain, there's then a corresponding net negative effect on S. And this is the basic concepts of immune cell regulation in our local tissue patch. Again, we have representations of 4 different cell death mechanisms. So 4 ways that epithelial cells can die. The first is virally induced apoptosis. And again, this is due to large amounts of intracellular virus that's in the stage of being what we've called assembled. So this is the last stage of viral replication inside a particular cell. There's the cell death due to the event of immune cells coming into contact with an infected cell. So if an immune cell does establish a contact interface with an infected cell, that infected cell then dies. Now, along with that event, we also consider a bystander effect, in which case, every time that contact cytotoxicity occurs, we then look at all of the neighbors of the cell that was killed, and then we assign a probability that each of those neighbors also dies, and that's our representation of the bystander effect. So neighbor cells killed by contact cytotoxicity also have a probability of dying due to the bystander effect. And then lastly, our 4th mechanism, we call it oxidizing cytotoxicity. Again, this is the case when an epithelial cell is exposed to a high level of our oxidative agent that's secreted by an activated immune cell. And in this case, the epithelial cell, irrespective of its type, dies. So uninfected cells and infected or virus-releasing cells all die, all under the same mechanism of high exposure to our oxidative agent field. Now, with all of that in mind, we'll show a lot of simulation results, and we'll also do a lot of simulations in the second half of our workshop today. And in all cases, we begin with a sheet of epithelial cells, all of which are uninfected. And those are all shown in blue here in the top subplot. And then we kick off the events of viral infection by infecting one cell. We choose the center epithelial cell in the entire sheet, and then we take an amount of virus and we put it in it. And then we hit run and we observe what happens. Now, along with that, we also initialize our immune cell population as being 0, which is why this is all shown as black. And then our virus, cytokine, and oxidative agent fields are also initialized all as 0. And for all of the simulations that we consider, we simulate 1,000 simulation steps, each of which is counted as 20 minutes per step. And this amounts to just under 2 weeks of simulation. Now, we began by finding what we call a baseline parameter set. And our baseline parameter set, other than educated guesses when possible, were mostly a guess. And we chose our specific parameters for our baseline parameter set to be at what we tend to call a sort of boundary of balance between when infection spreads throughout the entire tissue patch and when the immune response manages to contain infection and clear the virus. Now, to make things interesting, we chose our baseline parameter set to be on the infection winning side of the boundary. And so you can see with this time series of our 5 fields of interest, well, 4 fields. This is the same field. This is on a lower level. This is on a level above it. So our cell field, and then our virus, cytokine, and oxidative agent fields. You can see that over time, in our baseline parameter set, somewhere near the end of simulation, that everything dies. And we chose this near this boundary of balance between infection and control so that we could find model parameters of interest and vary them individually, and then observe what happens. Does death of everything occur much faster? Or is the virus contained? Or is spread of infection just slowed? In this case, everything dies, and we notice when observing the distribution of, say, cell death, that immune cells do a lot of killing by contact or secretion of the oxidative agent. And this is what it looks like. So this is the baseline parameter set. You see the cell population here. Now we're going to do this again with also our diffusive fields. So again, the blue cells are uninfected. The green cells are infected but not releasing virus. The red cells are virus-releasing cells, which you can sort of track in the virus field. And then all the black cells are dead. Now, to observe what the immune system actually does and how it affects the progression of events, even just with respect to our baseline parameter set, we did the same set of simulation parameters, except that we disabled the immune response. And this is shown here. This is the same sort of time series, but with no immune system at all. And you'll notice here that this is more of a much more uniform sort of Fisher type spread of infection, where much sooner than end of simulation, everything is dead. And so we notice from that, this is evident here, say when observing this purple line, this is the number of dead cells on a logarithmic plot. You notice this is the baseline parameter set, and everything died somewhere between, say, 17 and 18,000 minutes. Whereas when we look here for the reference case of no immune response, see that this occurred much faster at around 12,000 minutes. So even for the cases of widespread infection where everything ends up dead, even just by introducing an immune response in general, we see that there is at least this sort of delay in the progression of infection. Now we took our baseline parameter set and we found some parameters of interest that seem to have major effects on the progression of infection or its containment. The first parameter sweep that we did was doing parameter sweeps of coefficients that affected the delay in immune response, as well as the rate at which cells will internalize extracellular virus. So you'll notice here in these parameter sweeps, moving from left to right, the left columns correspond to a weaker, slower immune response. And then moving to the right, the far right column is the fastest and strongest immune response. Whereas going from the top row to the bottom row, the top row corresponds to much slower internalization due to exposure of the virus, and the bottom row is for very fast internalization. So our baseline parameter set is here in the middle. And again, we saw widespread infection. This is counting the number of infected cells in this plot and the total extracellular virus in this plot. And you notice in the top right, with the stronger immune response and the slower viral internalization, we get nearly a deterministic system. Each of these plots shows a simulation replica. For each of these parameter sets, we did 10 simulation replicas. You notice for these green regions that we mostly get, other than here, there's a bit of variation in this parameter set, but for all of these cases, we got no infected cells at the end of simulation. Whereas when we look at a much faster internalization rate and a much slower, weaker immune response, we get a deterministic behavior of very fast, complete spread of infection throughout the tissue patch. And all of those cases are shown here in this reddish region through here. And so we have these domains, these sort of like subdomains in the parameter space that we've chosen, where we get the same outcomes, whether good or bad. And then in between, in all of the unshaded plots, we have what I like to call a fuzzy boundary. And by fuzzy boundary, I mean that we can take the same parameter set, the same initial conditions, and then simulate it and find multiple outcomes. You'll notice here in the top left, when counting the total extracellular virus for these simulation replicas, we have some cases where at the end of simulation, the total extracellular virus is very high. And we look at the number of infected cells and we see that there are enough to say that infection is still occurring. Whereas when we look at the total extracellular virus for some other simulation replicas, we see that it's going to 0. And so for parameter sets like this, we see a lot of stochasticity and a lot of variation over simulation replicas. Now, we also noticed when really diving into these different simulation replicas and looking at what happened, that we could identify some sorts of classes of infection dynamics, where we noticed very similar outcomes occur, but interestingly also some dynamic events that occur throughout simulation. So these are shown here, and we'll just go through them very quickly. So there's the obvious case, like our baseline parameter set, which we call widespread infection, and this is the case when there are no uninfected cells at the end of simulation. Here's the case, the outcome of what we call slowed infection, in which case at the end of our simulation time, infection is still progressing. There are still some uninfected cells and also some infected cells. There's what we call containment. This is the case when we find at the end of simulation that there are no infected cells. However, when we measure the total extracellular virus, we find that there's an appreciable amount, meaning that even though there are no infected cells, that more infected cells could pop up. And so there's still the chance of infection starting again and needing to be dealt with. There's this interesting case that we call recurrence. You'll notice here when counting the number of infected cells, we'll find that at times there are no infected cells in the simulation domain. And then when we look farther in time, we find that there are infected cells that pop up again. And this is due to extracellular virus being in the extracellular space due to release of virus by infected releasing cells that had previously killed. and then the positive outcome of infection, which we just call clearance. This is the case where at the end of simulation we find no infected cells, no virus-releasing cells, and the total amount of extracellular virus in the simulation has gone down to below an appreciable level. So we then took our framework and looked to see how we could simulate something like a drug therapy. To actually simulate the course of events in infection dynamics, but then also introduce something like a drug intervention, and at some point during the course of events in time. So what we did was we took our viral replication model, which is shown here, and drawn this box around this term here in the replicating stage. Again, this is the term that brings about exponential amplification of the virus inside of a cell. Now, what we did in our drug therapy simulations was that we said that at some point in time, after infection, we will take this coefficient Rmax. This is a coefficient that controls the amplitude of this exponential amplification effect, such that if, say, we set it to 0, there would then be no replication. And we said that at various moments in time, we're going to reduce that coefficient Rmax by some amount, and we would do this at various times in simulation. Now we swept these two parameters. First, what we call our time delay of application. So this is the amount of time after infection has begun, at which point a particular drug therapy is applied. And this is shown from left to right. There's introduction of the therapy at the very beginning of simulation. To the right, this is very late after infection. And the other variation was the reduction in our coefficient Rmax. At the very bottom, this is the weakest reduction. The coefficients that were used are shown on the right here. So this is a 12.5% reduction from the baseline parameter of Rmax. Whereas at the very top, you see this coefficient says 0. This row corresponds to completely turning off viral replication at whatever moment in time during simulation the therapy was applied. Now we see something very similar to our first parameter sweep, which is that in the extrema, in this case the top left and the bottom right, we see nearly, in some cases, in other cases, exactly the same outcomes for all simulation replicas. Again, these are 10 simulation replicas per parameter set, and we see that for very early treatment and very strong treatment, that when counting the number of infected cells, the number doesn't really change. Whereas when we have very late infection, or very late therapy, and a very weak therapy, everything gets infected. Now, these measures are shown in green and orange respectively. So positive outcomes are green, and negative outcomes are this reddish-orange color. Now, there are a couple of interesting things to note about this. The first is that we observed that even for weaker treatments, say this next to bottom row, this is a 25% reduction in our coefficient Rmax. And we see that even for very early treatment, there's still a very strong chance of getting a positive outcome. Whereas when we have an absolute reduction in replication, which is shown in this top row, for very late treatment, see that there's still the possibility of negative outcomes. Now, the second thing of interest is, again, the emergence of our fuzzy boundaries. So between these sort of regions of deterministic outcomes, we then get all of this uncertainty and all these variations between our simulation replicas, even to the case, say here, where we have a clear bifurcation. There are some simulation replicas, again, the same initial conditions, the same baseline parameter set, and the same therapy. And in some replicas, the number of uninfected cells goes to 0, whereas in other cases, we have something like delayed infection, and significantly so. Now, one parameter set particularly of interest is what's boxed in here. We're going to take a look at that. These are 3 different simulation replicas of that same parameter set. This is shown here. And these are all of the metrics for each of those simulation replicas over simulation time. Now, we found for this same parameter set 3 different outcomes. This negative outcome being that there are no uninfected cells left at the end of simulation. This quite clearly is this purple curve. Whereas the positive outcome is quite clearly this green one, or something in between. Now, what we noticed when trying to explain these differences was that the reason for positive outcomes in the cases of drug therapy has very much to do with the stochasticity and its effects on very early infection. In this case, for our simulations, when we seeded our initially infected cell, we found that because of the stochasticity of virally induced apoptosis, we found that sometime after releasing virus, it died very early. But then recurrence occurred in a neighboring cell, at which case infection then progressed. So this is like delaying the onset of infection in simulation time. Now, for the case of drug therapy, this matters because this thing corresponds to an earlier therapy simply due to the fact that the first infected cell happened to die early. And this was the cause for the positive outcome. Now, the difference between the intermediate and negative outcomes is also particularly interesting because what we found was that when we looked at the number of cell types in simulation time and looked at the point when these 2 sets of events really started to diverge, we found that at around that time, the number of infected cells for each of these simulation replicas was approximately the same. It's about the same number of virus-releasing cells and infected cells, so on and so forth. The difference was the amount of extracellular virus in the simulation domain. Now, the only explanation that we can come up with for that is spatial effects. The difference between these 2 is the spatial effects of the nature of diffusion in the extracellular space of virus that's been released by virus-releasing cells, and where newly infected cells are positioned with respect to that initial site of infection. And so we conclude from that that stochasticity of cell-based events matter, as do the distributions of infection when considering the progression of an infection in local tissue. And this explains these sort of fuzzy boundaries between the deterministic regions that we observed in our parameter sweeps, where we got practically the same outcome over all simulation replicas. And then lastly, when simulating our drug therapy, we certainly observed that early treatment is better, which is perhaps not a surprise, but more interestingly, that early treatment makes low doses have a much better chance of being an effective treatment. Now, we can go to Nanohub, and hopefully those of you who have launched Nanohub have managed to find something that looks like this, or can be arranged like this. So this is our Nanohub tool, and this has been set up to launch and run our baseline parameter set. And this is what it looks like. So we see our first infected cell has now become virus releasing, and that's shown in red. We look in the extracellular virus field, and we see that there are viral particles now diffusing away from the first infected cell. Now, here soon, we should see newly infected cells near that first infected cell start popping up. So we have one infected cell there. You see that immune cells are being introduced according to our immune response state variable. So this has been positive up until this point, and as such, immune cells were added. Now we see more infected cells popping up in the epithelium. Soon we should see those go to red, in which case we'll see more sources of extracellular virus in the field. Now you see here our dead cells and the Nanohub tool are currently shown as yellow instead of black, and you see that the first infected cell has died, whereas some of its neighbors are now starting to become virus releasing because enough virus has replicated inside those first initially infected cells. And now from that, now that there are many more virus releasing cells, we should now see that our immune response state variable should start increasing drastically. You see here, immune cells doing contact cytotoxicity. This explains this patch of dead cells. There was an infected cell there, and it was killed on contact. Some of its neighbors were also killed by the bystander effect. You see that these two immune cells are doing quite a bit of killing through here. You see another instance of contact cytotoxicity and the bystander effect by this immune cell here. And now we also start to see that as the immune response state variable starts increasing, begins to see a drastic increase in the immune cell population. Now here, you see this massive patch of tissue damage, and also this group of immune cells here. Now this is certainly due to activation by cytokine signaling, and then release of the oxidative agent resulting in killing by the oxidative agent in sort of this more diffusive pattern. Yeah, you can see a lot of this now popping up. So at this point, these immune cells are now bombarding the epithelial patch with the oxidative agent. Should see now lots of cell death coming into play. Now, despite that, there's still lots of virus releasing cells here, in which case there's still cytokine signaling. More immune cells should still be introduced because this is positive. However, lots of killing just eliminated a lot of virus releasing cells, in which case we'll now see more of a decline in the immune response state variable. So yeah, we don't need to go through this completely for now, because we are going to spend a lot of time going through these simulations. So now I would like to stop talking for a minute and open up the session for some questions. So again, please enter your questions into the chat. We are certainly interested to hear any questions or comments about the model, any questions about how things have been constructed and put together, its conceptualization, and of course we also welcome questions that are specific to your research or things that we may be working on with our own model developments as well. So again, please, questions in the chat, and we'd love to hear from you.
If you haven't done it yet, now would be a great time to register for the Nanohub application so that you're able to work with it in the second half. And if you've decided to load CompuCell on your local computer, now would be a good time to go and do that and make sure that that's working.
Okay, so I'm scanning up a little bit earlier in the chat from when we were going through the model. I see a question here from Sean. Would the packing of the cells affect how the model progresses? For example, say hexagonal connectivity versus a grid. And yes, certainly. There's certainly a diffusive nature to the progression of the spread of infection, but there are also, as we sort of went through a little bit more, I think after the question was asked, certainly significant effects based on the position of individual cells. So certainly a hexagonal arrangement would have an effect. Certainly unfreezing the cells and allowing them to move would also have effects on the outcomes. Yeah, that's a great question. Question from Tom. Is it possible to account for repopulation of the tissue with new uninfected cells? Could this be run concurrently with the infection simulation? Like say to study the dynamics between viral infection and healing. Certainly, and this is certainly something that we've been interested in and have begun to start looking at. Yeah, some of that really depends on what sort of detail you'd like to include with a model of the epithelial population. So for example, the way I've been thinking of it, say we keep the epithelial cells frozen, then how do we replace a dead cell with an uninfected cell, given that you can't actually move the cells, in which case you'd have to do something like abstracted some sort of description of how a particular dead cell then gets flipped back to being an uninfected cell. It's certainly not like resurrection by any means, but more like saying that in this space where that dead cell was, that something happened to replace it with a new uninfected cell. On the other hand, assuming that you were to unfreeze the epithelial cells, then certainly when a cell dies, you would do something like removing it, actually removing it from the spatial domain, in which case there's space available for proliferation, in which case can either introduce a new cell into the space, similarly to how we do immune cell recruitment, or you could actually do division by, say, a neighboring cell that senses that there's available space. So just some very elementary ideas on how to do that. Okay.
TJ, maybe you could actually show them the Nanohub, how to do the tool launch and sign in, because I think that would be helpful at this point.
Okay. Through the entire sign-in process?
I think if you go to the tool webpage, you hit launch tool, then people will be asked to create an account, and then they can go back to that page and launch the tool.
Okay. So using the link, go to here, and then launch tool. If you've not created an account, you can go through the registration process very quickly. This will then launch the Nanohub tool. It'll be a bit less organized than how it looked when I first demoed it. You'll notice that it starts as running, so you can pause it by hitting the pause button. If it's not centered on your screen, you can move it over and maximize it. I prefer to close the model editor for more space.
You can also get more space, TJ, if you scroll down a bit. That arrow with the dimensions, you can drag there to make it bigger.
Okay. Thank you, Giuliano. And then to organize the workspace, you can click on any one of the window panes, and then go to Window and Tile. And then to look at what we'll mostly focus on today, this is initialized with the 2 layers of the cell field and the cytokine field. You can change results for a particular window by clicking on the drop-down menu and selecting a different result, say, in our case, the virus. And also in our case, you can go up and down the levels. For our cases, we'll mostly focus on the virus field at the level of the epithelial cells. So that's how to put together the CompuCell player, the way it was arranged during the demo. Okay, I think I saw a few more questions. And again, if I missed any, please just drop the question into the chat again. Let's see. Question from Rory. Has anyone looking into hormone receptors and gender differences? Yeah, so that's certainly of interest. You know, James knows much more than me about who all is looking into what, but there's certainly a lot of interest in thinking about differences in these sorts of terms. We're certainly interested in differences in sex, in age, ethnicity, and so something more like hormone receptors is certainly something that we can like really abstract in terms of mechanisms that we model, whereas something like a difference in sex, that's, you know, that's a little harder to conceptualize in terms of simple mathematical models, and instead probably show up in differences in, like, say, parameters, or you know, location that's being simulated. So James, if you'd like to weigh in on that, I know you certainly have a lot to say on those sorts of differences, and we have plenty of collaborators who we've been talking to that are interested in these differences as well.
Well, certainly things like ACE2 receptor are differentially expressed. Some of the immune responses are differential. And so with time, we would like to include those. As you say, they'll typically show up as parameter differences in terms of differences in inflammatory response, differences in populations of immune cells, and in receptor densities. And so anyone who has suggestions about particular data sets that they'd like to see replicated in these kinds of models, we'd be delighted to talk to you about
Okay, and then I think, so I see, so there's a question from Maria. Should I start learning Python to create a model about infection pathogen? So a lot of that depends on how detailed you really would like to go. And we try very hard to develop CompuCell in such a way that there's a very wide range of involvement in detail that you can really go, staying as either high level as you'd like or really diving into the weeds. So if you have, say, some particular mathematical models that you've been playing with, as say ordinary differential equations or describing certain cellular or subcellular mechanisms, Python in CompuCell is the way to do that. We try to make it as easy as possible. Python is certainly one of the easiest languages to work with. We provide a lot of utility functions and a lot of support for doing model specification, hopefully in an intuitive way. So if you have actual models that you'd be looking to develop, then Python in CompuCell would be the way to do it. But even doing development to start in Python is very straightforward. I would say extremely straightforward. I mean, we provide a lot of manuals and documentation on how to both use the framework that we provide, as well as doing basic computations and things of interest for bio complexity modeling. So Python is yes, the place to go if you'd like to actually develop specific mathematical models. Okay, question. How easy is it to match parameters to a specific virus? Would it be possible to distinguish between influenza and SARS-CoV-2? This is my favorite question right now. So we say that we can. We have cases in the past, previous work, where we've been able to calibrate model parameters to specific events with experimental data. And currently, for me personally, one of my specific interests right now is to take this simulation framework with all of the model parameters and take these sorts of global measurements, not global in the sense of like the body, but these are scalar metrics of spatial data in our simulations, and take those values and use the simulation framework as feedback in a sort of something like an artificial intelligence supported workflow, and find the model parameters that produce these transient metrics consistently with a set of experimental data. So we know that we've done it in the past. It's not easy, and we're currently interested in exploiting artificial intelligence and optimization algorithms to find the model parameters that reproduce data. We would like to think that if we got a workflow like that developed well enough, that it would be as straightforward as giving, say, a software package a set of experimental data and a simulation framework, and then just tell the workflow, find the parameters that make the framework do what the data says. That would be one of our long-term goals. Okay, so I hate to cut this short, but it is, what time is it right now? Yeah, so we're starting to get behind. So for whatever questions we haven't gotten to, if you could just save those, we'll come back to the end and we can revisit the remaining questions. I'd like to take a quick break and let everyone stretch their legs, go get some water or coffee for those of us who are still in the morning time. Also, this is sort of the last call chance to preferably launch the Nanohub tool and get that going. But for those who prefer local installations, you can find those here. Again, the links, I'll drop those into the chat one last time. And then when we come back from the break, then we'll start getting into actually firing this framework up, playing with model parameters, and seeing what interesting things will happen just based on simple variations. So let's say we'll meet back at 10:35.
And again, I'd encourage you, if anyone is having trouble either getting into the Nanohub app or downloading onto the your desktop machine for the local CompuCell installations, please do ask us now. We'll put you in a breakout room with somebody who's an expert and make sure that when we come back in 5 or 10 minutes, everyone can run the code and see what happens as you change the various simulation parameters. There have been a lot of questions about how to extend the code to include new cell types, new chemicals, particular cytokines, questions about whether we can model infection of the immune cells themselves. Those are actually not difficult to implement computationally, although you have to understand the biology of what you want to do. And we'll show you how to do those kind of extensions next week. This week, the goal is to play with the existing code, which you can already do quite a bit with, and then to think about what the most important extensions that you'd like to have would be, so that we could work together to build a simulation that represents the immune system response and tissue damage in a way that is meaningful from the point of view of all of it. Someone asked a question about academic backgrounds. This is James Glazier talking again. I actually trained as an experimental physicist many years ago at the University of Chicago. I did a postdoctoral work in experimental neuroscience, and then did my early work as a faculty member primarily in experimental developmental biology, became interested in computational implementations of developmental biology mechanisms. And that's what led to the development of this CompuCell3D software environment. And we've worked primarily over the years in what we would call developmental diseases. Those would be diseases that deploy developmental mechanisms in pathological ways. Cancer is the most famous of those, but diseases like polycystic kidney disease or diabetic retinopathy are also developmental diseases. TJ, who's giving the presentation, has a background in mechanical and bioengineering and also computer engineering. Joshua actually comes to us from philosophy of science originally, history and philosophy of science, but now is doing a PhD in bioengineering. Giuliano has a background in solid state physics and computational modeling. Jim Sluka, who's on the call as one of the facilitators, has a background in biochemistry and toxicology. And we also have a variety of experimental developmental biology and toxicology and now virology and immunology collaborators. Philosophically, one of the things that I believe is that biology is difficult and complicated. And so we need to make the implementation of biological concepts in computing as simple as possible. Really, we want to be able to make it so that experimental biologists, clinicians can build their own models because they're the ones who understand the biological processes at work, and to minimize the amount of computing that's required to take biological ideas and translate them into meaningful computer models. We definitely encourage you to contact us outside of the workshop if there are questions about CompuCell, about modeling any biological system, or specifically opportunities or questions about modeling COVID-19. We're always eager to find new collaborations and to help people apply these methods in their research, whether that research involves immunology and virology or something totally different. If there are any other questions in the meantime, we're happy to take them and answer them to the best of our ability.
Okay, so let's go ahead and get back to it. Start working on getting CC3D fired up. So here are the links that were posted into the chat. Since we're running a little bit behind, I'm just going to assume that everyone has gotten their platform of choice running. Again, Josh and Giuliano are here to assist for anyone who might be having issues. I'd also like to say that if you are having any issues with a local installation, we would certainly be happy to work with you after the workshop to get your local installation working, and would advise instead to switch over to Nanohub, as all of that is configured and platform independent. So this will certainly be sufficient for the rest of the workshop today. And then again, Giuliano, Jim Sluka, and I especially would be happy to work with you individually after the workshop to get your local machine running CompuCell as well. But for those of you who are using the local installation, you can find the simulation files updated live as we develop them from GitHub. There's a link in the chat to our GitHub repository that is updated regularly. We always keep our work open source and public, so you can check in on that regularly and see not only the current state of the framework, but every state that it's been in since its conception. Also dropped a link into the chat to the Google Drive simulation files, so you can find the simulation files here as well for those of you who are more allergic to GitHub. If you have downloaded the GitHub repository, which you can do here, you open the GitHub link and download the zip. You'll get one directory of the entire repository, and you can find the simulation files in this directory called model. Now, that being said, let's start with Player. So for those of you who launched the Nanohub tool, Player opens automatically. Otherwise, for those of you with a local installation, you should be able to just type into your start menu CompuCell, and you should get a shortcut to CompuCell Player. And when you launch CompuCell Player, it looks like this. Again, the Nanohub tool will launch with the simulation files loaded and running. So you'll start somewhere like here. Local installations will start here. Now, to get the simulation files loaded in Player, we'll go to File, and then Open Simulation File. And when you navigate to the model directory where you downloaded your simulation files, you should find a file called viral-infection-vtm.cc3d. When you find that, select it and click Open. You should notice in the window title for player that the file name viral-infection-vtm.cc3d should show up. Excuse me. Now for the case of Nanohub, you can do the same for loading this simulation script or others by going to File and then Open Simulation File. Now in Nanohub, you'll find that in this left pane, you should have a directory with your Nanohub username. And when you select that, you should find to the right a directory called CompuCell3D _demos. When you open that, you should find a directory called coronavirus. Select that. And this will take you to the simulation files. You can just select viral infection vtm.cc3d and open it. And this loads the simulation file for a model specification into player. Now, the second main software that we distribute for model specification with CompuCell is called Twedit. Twedit is a text editor with lots of built-in features for code snippets and so on and so forth, and the XML and the Python to facilitate doing various commands and functionality. So you can launch Twedit from a local distribution or in Nanohub from player with this button here. When you highlight it, it should say Start Twedit, and when you click it, it starts Twedit. Now in Twedit, you can load an entire CC3D project similarly to how we did it in player. In this case, look in the file menu bar, or I'm sorry, in the Twedit menu bar, see menu item CC3D project. When we select that, we find the menu item Open CC3D project. When we select that, we can navigate to the same location with the model simulation scripts and find viral infection vtm.cc3d. When we select that and click Open. And this is the same procedure on Nanohub. Twedit will launch in Nanohub as well. And again, CC3D project, Open CC3D project. And then again on Nanohub, you'll find a directory with your username on the left. And then open CompuCell3D demos. And then open the directory coronavirus. And then select the CC3D file viral infection vtm.cc3d. And open. Now, in both the local installation, as shown on my screen, and on Nanohub, should have Twedit open with a blank document, and on the left, we should find a selectable item, viral infection vtm.cc3d. So let's double-click that. Now, what you should find is that a lot of simulation scripts opened. And in Twedit, these will show up as different tabs along the header. And you can select each of these and they'll open these individual scripts. These are all of the simulation files that specify the entire framework for the viral infection simulation. Now, we're only really going to deal with one of these today, do a tiny little bit with another. But the only file of interest for our current purposes is the file called viral infection VTM model inputs.py. And this simulation script is specifically dedicated to defining all of the model parameters, in this case, quite a few. When you go to that script, viral infection VTM model inputs, and scroll all the way down to the bottom, see 291 lines specifying lots of model parameters, what their values are, and then some descriptions of what they do. Now, all of these parameters are the baseline parameter set that we previously reviewed before the break. And what we'll do today is we will do individual and sometimes paired variations and select model parameters and see what they do. Now, let's go back to Player. Hopefully by now, everyone has viral infection VTM.cc3D loaded in Player. Now, when we look to the top left of Player, you see control buttons to play, step, pause, and stop a simulation. So let's start, for anyone who hasn't started a simulation, if you have, you can hit stop here. And let's start by selecting the second button, which is called step. Now, the simulation framework, or really the simulator in CompuCell, moves through time in simulation by doing individual time steps. We call them just simulation steps. And what these first two buttons do, the button run, the first one, will just step through simulation until you either pause it or stop it, or it'll stop at the end of simulation. Whereas the second button, step, does exactly one simulation step and then pauses. Now, depending on your installation, or if you're using Nanohub, you may have gotten all sorts of different results. Panes, we went through the organizational process in Nanohub before the break to sort of organize some of these, because there are a lot of results that can be reviewed. Player will render results in real time according to the window panes that you create and the results that you specify for that particular pane. So for our intents and purposes, we'll mostly review the two levels of the cell field as well as the extracellular virus field. So you can create a new results pane by selecting this button in the player, new graphics window. And you'll notice that when we select that, well, this has to be running to do this, mind you. So we can select step to make sure that the simulation is loaded. And once the simulation has loaded, you can then create new graphics windows with the new graphics window button here. See that when I selected that, created this new window here. And then from there, you can use the drop-down menu and select a results field of interest. Say, the virus field. Now again, this model is a quasi-2D simulation. We have a planar epithelial sheet, and then above that there's a cell field of, not a cell field, I'm sorry, above the epithelial sheet there's another level where immune cells are populated. And we can move up and down that coordinate using this menu item here. You notice that this says 1. The coordinate z equals 1, since we're looking at the xy plane. Z equals to 1 is where the immune cells are. You see that there are none there currently. Whereas when we go down one coordinate along z, we find the epithelial cells. So you can create whatever graphics panes you wish, and that might interest you. I will be working with the 2 levels of the cell field, as well as the virus field in the level of the epithelial sheet. Now you can organize all of your graphics panes into something nice and neat by going to Window, and then Tile. And you should find the player will organize these into a nice grid so that you can see everything that you've created in a nice coherent way. Again, this is all the same in player. On a local installation, so the button is the same to create a new window pane. Again, this only works if a simulation is actually either running or has been stepped. And you can go to Window and Tile. Now, what we're going to do is we're going to take individual parameters and make modifications to them from the baseline parameter set, and then we're going to see what happens. So we've set this up to facilitate this as much as possible to try to stay out of the code. Josh and Giuliano have set up what we call a steering panel, and this has been programmed such that we can select one of these drop-down buttons for an entry. And when we select one of these available items, this will apply a multiplier to the baseline parameter. So when all of these say 1, then there are no changes. Whereas if we say select 10 for what's called k-on multiplier, then the corresponding parameter k-on, which we'll get to in a moment, will be multiplied by 10. And this is done in real time. So you can actually make these changes midway through simulation, and the simulator will update those parameters according to the configuration that you specify here. So we will start by playing with the rate of infection, specifically with doing a variation on this parameter k-on that affects the rate of internalization for a given exposure to extracellular virus. So you'll notice here this is an expression for the probability that uptake, internalization of virus into a cell from the extracellular space is modeled as a Hill equation, where the dissociation coefficient is affected by this coefficient k-on, which is a coefficient that models the association affinity between the virus and a cell receptor. Now the sensitivity is such that if we were to increase k-on, then there's a corresponding increase in the rate of internalization. So let's try that. We go to player, let's start with a stopped simulation. Now, if we hit step, this will both load the simulation file and do the first step. And we can do window tile to clean up again. Now from here, again, the simulation is paused because we only did step. So we're at MCS, or just simulation step 0. And now let's apply a multiplier to K on. Let's increase it. Let's multiply the baseline parameter for K on by 100. This should increase the rate of viral internalization. And what we should see is a faster propagation of the infection through the tissue patch. Let's hit run and see what happens. So on my screen, I am at 50 simulation steps. I have one virus releasing cell, excuse me, and much faster propagation of the infection front. In fact, before 100 simulation steps, I already have many, many more infected cells, a much faster propagation of the infection. We can watch and see how this plays out and see how the immune system deals with it. So I see some patches of dead cells popping up from presumably contact killing due to the presence of some immune cells. But otherwise, a mostly sort of diffusive expansion of the infection front throughout the tissue. But at this point, notice in one of the graphics panes showing the state variable S for the immune response, but at this point, the state variable is increasing a lot, in which case, when I observe the immune cell population, the more immune cells are being added. And then looking back at the epithelial cell population, with that increase in immune cells, I now find this massive area of cell death. And in fact, when watching them, saw quite a few patches of dead cells pop up very quickly with a very diffusive nature, which very much implies death by secretion and exposure to the oxidative agent by activated immune cells. I certainly see a much thicker rim of infected cells at the front of infection, and my immune response is skyrocketing. Now, if you see something different, please include it in the chat, because there are lots of stochastic elements to this. And when we see a variation between simulation replicas, it's always of interest. We certainly want to understand both what can emerge from a parameter set, as well as why. I would assume, though, that most everyone is seeing this outcome, which is a very rapid spread of infection throughout the patch. Now, I am at almost simulation step 500. I'm going to pause when I first see the last uninfected cell get infected, which is not for me. And that was at simulation step 484. Now, I'd like to revisit the fact that in our baseline parameter set, we saw widespread infection somewhere nearer to the end of our simulation, which was 1,000 steps. Now in this case, with this increase in K on, the corresponding increase in the rate of internalization, at simulation step 484, much, much sooner than 1,000 simulation steps, we see widespread infection. That is a massive increase in the propagation of infection just due to an increase in the representation of the association affinity between the virus and cell surface receptors. Now, let's do the opposite. Let's stop our simulations and let's do a variation in the opposite direction. Let's stop and then press step to restart a new simulation and to pause after the first simulation step. Now back in the steering panel, let's go back to our K on multiplier. And let's reduce the K on multiplier to a value of 0.1. So this is the same as dividing the baseline parameter by 10. And let's hit run and see what happens. You should see no differences in the behavior of the first infected cell. Have release of the virus by the virus releasing cell. If you happen to have a graphics pane suddenly go black, this is a bug that's been plaguing me for months in CompuCell. You can just close that graphics pane, create a new one, and reassign your same result of interest and location. You've done nothing wrong. It's just a bug that I've not been able to find yet. Now, I'm at 140 simulation steps now. I certainly see progression of some infection. But also, my first infected cell has already died. Now, no immune cells nearby, which would mean that that first cell just happened by chance to die early due to virally induced apoptosis, which we saw in some of our stochastic outcomes before the break, which led to one of the positive outcomes in our drug therapy simulations. Now, even though that cell died, all of its neighbors by now are either infected or worse, virus releasing. We see new sources in the extracellular virus, where those cells are now releasing into the extracellular space. We certainly see that it's not nearly as rapid as what it was. On my screen, I am coming up on 350 simulation steps, and I see certainly a spread of the virus, but not nearly as rapidly. See lots of cells being killed by contact cytotoxicity and the bystander effect. Now I see much larger areas of cell death popping up, indicating secretion of the oxidative agent and corresponding cell death. And then these interesting cases, much farther removed from the main body infection, where individual infection sites have popped up. This is purely due to the nature of diffusive transport of virus that's been released, and then stochastic evaluation of internalization at each of these individual cells. Now I'm halfway through this simulation at 500 simulation steps, and in my replica, I have lots of cell death, and I certainly have some infection still progressing, still being worked through by the immune system, but certainly not the massive, rapid, widespread infection that we saw when we varied K on by a factor of 100. In fact, in this case, it very much looks like by the end of simulation, with this one variation, that this will likely end up being something more of a case of slowed infection. So we see lots of cell death happening. We certainly see progression of the infection, but finding this large rim of infected and virus-releasing cells, as in our previous variation, is certainly not apparent here. Much more sparsely distributed, much thinner rims of virus-releasing cells. You can see that this reduction gives the immune system a much better chance of dealing with infection and containing it. You can certainly see some of the differences in timing and the spatial effects when we look at, say, the amounts of virus in the extracellular space. You can look at these regions of virus-releasing cells to the left or to the right, in my case, and you'll notice that the amount of extracellular virus is different in those different areas, and that's purely to do with the timing and the amount of internalization for each individual cell. So this is clearly the effects of spatial, temporal, and cell-based effects. Okay, so my simulation is stopped, and I would guess, based on my outcome, that widespread infection will occur, and that it's going to continue to spread, albeit much slower, but certainly a difference from the baseline parameter set. And we would then extrapolate from this that something like an adaptive immune response that would come in later is much more specialized to dealing with this particular pathogen. But the adaptive immune response would have a much easier time dealing with an infection like this with a decreased model parameter K on. And so these are the sorts of corresponding higher level effects that we would expect to see. We see a reduction in the rate of internalization. In the very least, something like an adaptive immune response would have a much better chance of dealing with an infection. So the second parameter that I would like to play with a bit controls or affects the amount of delay between the local signaling of the cytokine, the local signaling of infected cells via cytokine, and then the corresponding response of the immune system and infiltration of immune cells into our local tissue patch. Now, this one will require a minor modification to the code for those of you who are using Nanohub. So for those of you who are using a local installation and may have downloaded the Google Drive simulation files. I patched an issue this morning that I found in our previous session yesterday. And we're going to fix that very quickly so that we can play with this recruitment parameter. Notice here in the steering panel that there's a second item, beta delay multiplier. Our Nanohub implementation is missing one line of code to make that work. So in Twedit, let's go to Twedit, and along the top, you can find the file viral infection VTM steppables.py. Now, let's go down towards the bottom to line 907. Now, what you should find is a line that looks like what I'm showing on my screen. And if you don't, then you'll need it. Nanohub users won't find it. Those of you who downloaded a Google Drive version of the simulation files should find this command. Now, I will paste this line into the chat now. So you can copy the line that I just dropped into the chat. And I'll walk you through doing it, say, on Nanohub. So on my Nanohub tool, when I go to Twedit and viral infection VTM steppables.py, down to line 907, my Nanohub tool doesn't have that line. So I'm going to add it. Except I can't in Nanohub. Giuliano, can we not paste things directly into Nanohub? Or James?
Sorry, it's not allowing you to do a cut and paste into the document?
It is not. I don't want to bog everyone down with doing a bunch of coding. So if we can't paste, that's fine. We'll just move on. I don't think we can. Okay, that's no problem. So let's skip these. Let's look at a different one. I apologize for the confusion. So the next thing that we'll look at is one of the mechanisms that we're particularly interested in, which is genomic replication of the virus once it's inside the cell. So in the model, we have this exponential amplification term for the replicating stage of the virus. And this is where one unit of virus makes more units of virus. And what we'll do is we're going to do variations in this coefficient Rmax, which we call a couple of different things. For now, we'll refer to it as the replicating rate. And what we should find is that when we increase this coefficient, we should find that the virus replicates faster inside of a cell. And if we decrease it, that the amount of production of new virus decreases. Now, this has been set up and tested in the steering panel. So in the steering panel, in the third entry, you'll find Rmax multiplier. So let's start by increasing the multiplier for Rmax from the baseline parameter set. So for anyone who has a simulation that's currently running, you can hit stop. And then let's click step to restart with a new simulation. Now let's go to our steering panel once the simulation is fired up and go to our entry for Rmax multiplier. And let's start by decreasing it. Let's decrease it by a factor of 0.5. So let's select 0.5 for the Rmax multiplier. This is dividing the baseline parameter by 2. And let's see if widespread infection still occurs. So let's go to the top left and select run and watch it go. So for my simulation, I still see infection, see release of the virus by the initially infected cell. And then my initially infected cell died early. Now, the neighboring cells, in my case, from the initially infected cell, those are now all virus releasing. I see lots of spread of infection and corresponding increase in the extracellular virus field due to viral release. I see immune cells arriving. See a large patch of dead cells growing, certainly with diffusive qualities. So likely from oxidative agent secreted by activated immune cells. Now I'm at 300 simulation steps, and the immune system even hit a point of becoming anti-inflammatory. Very briefly, but for a moment, the infection was mitigated enough, still going, but was mitigated enough that at one point the immune system decided to actually remove some immune cells. Now, certainly infection is still progressing. There's still virus being released, but not a lot of progression from what managed to survive the first initial immune response. The second thing, on my simulation replica, perhaps some of you are seeing the same thing or have seen it in previous replicas, far from all of the activity near the initial site of infection, all of the cell death and so on and so forth, and now happen to have two uninfected cells, mostly I would say sufficiently far away from the main activity, become infected. Now, this is permissible because transport of the virus, once released, propagates out symmetrically and spherically. So it emanates away from sites of release. And though when we look at the contour fields for the amount of virus in the diffusive field, it looks as if there is no virus far away from sites of release, when in fact there is virus, just not very much. And when there is virus, even at very small amounts, it's still possible for individual cells far away to become infected because there is still some degree of exposure. Now, for cases like this reduced rate of replication, the main location of infection now is mostly docile, not very aggressive at all, and there's not really been much change. And then when I look at my immune cells, I'm at about 700 simulation steps. A few have just been introduced, but by and large, the immune system has not been very responsive. And the reason is because the amount of virus inside of a cell affects the amount of cytokine that an infected or a virus-releasing cell secretes for immune recruitment. Now, since the replication rate has been reduced, the amount of virus in each cell is not going up nearly as quickly, which means there's much less immune recruitment signaling, and therefore fewer immune cells. By and large, doing a reduction like this, decreasing the replication rate by a factor of 2 from the baseline parameter set, by and large, it certainly makes this infection much less aggressive, to the point where I would even go as far as to describe the immune system as being mostly indifferent to it, somewhat unthreatened, as it were. Now I'm coming up on the end of simulation for my replica, and we would classify this as something like slowed infection, but it's certainly not slowed infection in the same respect as our previous simulation replica, where infection was clearly still propagating and most cells had at least been infected, but not all. This is clearly slowed infection in the sense of that this particular represented virus is not particularly threatening. Now, let's do the opposite. Let's take that same parameter variation, and instead of dividing by 2, let's multiply by 2 and see what happens. Now, you'll notice in the steering panel, when you click on the drop-down menu for the item Rmax multiplier, notice that all of the coefficients that have been programmed into here, these are all for reductions. All of these will reduce the Rmax multiplier by a certain amount. You see there's nothing greater than 1 though. So to implement an increase in the Rmax multiplier, we'll have to go into TWEDIT. So for anyone who has not currently launched TWEDIT, again, you can launch it with this button here in the menu bar. Now when we go to TWEDIT, let's go to our model inputs script. So this is viral infection VTM model inputs.py. And now let's go down to line 145. It should look like something like what I have on my screen currently. So again, this is all programmed for the baseline parameter set. Now if we want to multiply this baseline parameter value for the replicating rate by 2, we can just go to the end of line 145, and then we can enter times 2. Now this is a programmatic modification to the code. You'll notice in Twedit that the little disk, a little floppy disk looking icon next to the model or the script file name has turned red. That means that it needs saved. So you can do any programmatic changes to these scripts and then save it project-wide. So this button here in the top left, the second button, Save CC3D Project. This will save all of your changes. On Windows, you can do Control-S as well. Now, when we go back to Player, and we restart with a new simulation, all of our changes will be reflected in the new simulation replica. So after having made our modifications in our scripts and then having saved all of them, we can select Stop in Player if anything is running. And then now we can just hit play or run. So let's do that. Stop if anything is running, and then after having made the modifications and saved them, we'll just hit run. And what we should see, after having doubled the replicating rate, the amount of virus being produced by a particular infected cell should be increased by a factor of 2, in which case we would expect that the rate of infection as it propagates throughout the tissue patch should be noticeably bigger, should spread much faster. So let's see if that happens. Let's select run. On mine, you should all see the same. In the baseline parameter set, the first initially infected cell would typically go to virus releasing state at around 30 simulation steps. Mine was there by 15. And now mine is already dead. Since there are no immune cells nearby, presumably due to virally induced apoptosis. I also already, before 100 simulation steps, have 3 virus-releasing cells. They then died very rapidly thereafter. Certainly see infected cells becoming virus-releasing cells much faster, and then certainly see virus-releasing cells dying much faster. And you'll notice that when far away from the immune cells that may be nearby, you should still see those virus-releasing cells dying very quickly. And that is certainly due to the extra amount of virus inside of a particular virus-releasing cell due to the much greater exponential amplification of the virus inside of that particular cell. So my replica is coming up on 300 simulation steps, and could already confidently predict that this will lead to widespread infection. See cells dying very quickly, even at the sort of front of infection, very soon after they become virus releasing. So clearly the death mechanism in this progression of damage is due to the effects of the virus itself. Certainly, this increase in replication increases the ability of this virus to propagate throughout the tissue, which we would expect. Now, again, the baseline parameter set exhibited widespread infection somewhere, say, between 800 and 1,000 simulation steps. In my particular replica, I paused it at approximately where the last uninfected cell became infected. And in this case, I measured that at around 425 simulation steps. So as we hypothesized for this parameter variation, the increase in the exponential amplification, in the increase in the rate of genomic replication, should be a corresponding increase in the progression of the infection throughout the tissue patch. Certainly what we saw here. So we can hit stop on that. Or of course, you're welcome to continue observing what that does from there. Now, there's another interesting mechanism, one that we were just observing with the increase in replicating rate, which was cell death by the effects of the virus inside of the cell. So we said that when we increased the amount of virus inside of a cell and we increased the rate of its production, that we would see cell death occur much sooner due to effects of the virus. Now, the expression that models that is shown here in the top right of my screen. So this is a stochastic function that says that the probability of a virus-releasing cell becoming dead is a Hill equation of the amount of assembled virus inside of a cell. Now, as we observed in the last parameter variation, we increased the amount of replication. Cell death occurred faster due to the virus. This would be due to a faster accumulation of the assembled state of the virus inside of a cell. And this is the expression where this is implemented. This would be why we saw that increase. This is the assembled virus inside of a cell. Now, there's a model parameter in this Hill equation that affects the probability of cell death due to assembled virus inside of a cell, which we call the Apoptosis Dissociation Coefficient, which is shown as V in the equation, V apo. Now, when we increase this dissociation coefficient, a particular cell will require more assembled virus to die due to the presence of the virus. And likewise, when we decrease this coefficient, the cell will die due to less amounts of internal virus. So let's vary that and let's see what happens. So let's go back to Twedit. And if you made the previous variation, then you should still be at line 145. You should still see this ×2 multiplier that we implemented programmatically. So let's remove that modification and set the replicating rate back to the baseline parameter set. Now to find the value for the dissociation coefficient that we'll modify, you can go to line 255. You should see this variable that would read something like dis coef uptake apo. It's a variable that equals 100 at line 215, excuse me, line 215. Now, again, if we decrease this, we should see that cells die much sooner due to viral effects. So let's take this parameter at line 215 and let's divide it by 10. So at line 215, we will take this baseline parameter that was 100, and we'll just add to the end divided by 10. And now let's save. Now we can go back to player. If we have a simulation running, we can hit stop to restart it with our new parameter values. And now we'll just hit run. Now this is going to load and run our simulation specification after dividing the dissociation coefficient of apoptosis divided by 10. Now I would expect to see that this first infected cell should die pretty quickly, though it's, so mine died at around 55 simulation steps. Again, due to this reduction in the dissociation coefficient for apoptosis. However, it did manage to release some virus into the extracellular space, and so at some point, you should still then see new infected cells show up. You should see the infection of uninfected cells, and this will be an event that we've named recurrence. So at the moment, the infected cell that we started with is dead. I have extracellular virus and no infected cells at the moment, but would expect that there would be some internalization at some point due to all of that exposure from release by the first infected cell. Of course, these are all stochastic simulations, and so there's always the chance that what I'm looking for, or what I think will happen, doesn't happen. And then I restart, and then find what I'm looking for. At this point, at least in my simulation replica, this looks like what I'm looking for isn't going to happen. Now, to check and see if this just completely stops spread of infection, I'm going to stop and run again. If you have infection progressing, then by all means, please continue observing your replica. I need to restart mine and see if I get the same thing twice, which is a complete failure to infect. Just very premature death of the first infected cell, and then no effect thereafter. So my new simulation replica has my initially infected cell still alive at 70 simulation steps, is now dead before 80, but has managed to infect a nearby cell. Now my replica is at 150 simulation steps, and I still only have one infected cell and one dead cell. Everything else is uninfected. This is pretty boring. So this premature death due to viral effects seems to severely inhibit the ability of the virus to propagate throughout the tissue, because in this case it kills its hosts way too soon before it can make a sufficient amount of replicas of itself to propagate. In my case, I am coming up on 300 simulation steps, and I now have one infected cell and one virus releasing cell. Very boring. So now I would like to do a variation in the opposite direction. Let's stop our replicas in player by hitting stop in the top left. And now let's go back to Twedit. And let's take our variation of dividing by 10, and let's change that. Instead of dividing the dissociation coefficient at line 214, instead of dividing it by 10, let's do an exercise in absurdity, and let's multiply it by 10,000. So the dissociation coefficient at line 215, we're going to multiply this by 10,000. Now this is an absurdly high dissociation coefficient compared to the baseline parameter. And we would expect, if we're considering our scales appropriately for this parameter, we would expect that this, in effect, will disable virally induced apoptosis by taking this value something like a limit towards infinity. So let's make our modification of multiplying by 10,000 at line 215. Now let's go back to player for any replicas that are running. Hit stop. Be sure to have saved your modification in Twedit before running this in player. And now with a saved simulation script with the modification and a stopped simulation in player, let's hit run. And what we should observe here is cell death only due to the intervention of immune cells. Again, increasing the dissociation coefficient as extremely as we did effectively should turn off virally induced apoptosis. And now whatever cell death we observe in the epithelial sheet should only be due to contact cytotoxicity, corresponding bystander effect, and then for activated immune cells, the release of the oxidative agent. So my replica is coming up on 150 simulation steps. My initially infected cell is still very much alive. All of its neighbors are now virus-releasing cells. I see some cell death due to contact cytotoxicity and the bystander effect. I have a very prominent core of virus-releasing cells at the center of this propagating wave of infection, and then immune cells that were recruited. See diffusive cell death near a population of immune cells, very likely indicating oxidative agent killing. I'm coming up on 300 simulation steps. I have lots of immune killing, both by contact, here, bystander effect, and then sudden appearance of large areas of cell death due to oxidative agent. And by 400 simulation steps, I have yet to see any indication of virally induced apoptosis. Of course, can't show this quantitatively without actually looking at the numbers, but I've yet to observe a moment where I saw cell death and then looked at the epithelial layer, or the immune layer, and found no immune cells nearby. So of course, we've been adding improvements recently to do better data analysis of these simulation replicas. And one of the tools that we've added that we will look at for our session next week is actually tracking the mechanism of each individual cell death. So with modifications and improvements that we do on our next session, we'll be able to do exercises like this and look at how we're affecting cell death mechanisms, but quantitatively by tracking the total number of cells killed by each of the individual cell death mechanisms. Now, my replica is approaching 650 simulation steps. I'm going to pause again when I see the last uninfected cell become infected. And I measure that at around 681 simulation steps for my particular replica. Now, if we place widespread infection for the baseline parameter set at somewhere between 800 and 1,000 simulation steps, we certainly see an increase in the rate of viral replication, or the propagation of infection, by comparing, say, those results to here, where widespread infection occurred before 700 simulation steps. But we certainly don't see the rate of increase in the propagation of infection like we saw when, say, increasing the rate of viral replication. in some of our previous exercises, I observed replicas on my machine with widespread infection closer to 500 simulation steps or even less. And so certainly see how disabling virally induced apoptosis contributes to the spread of infection. But there are certainly other mechanisms that, for the right parameters, contribute more. Okay, so I'm going to stop my replica. We have 10 minutes left on our time today. So I would like to finish this out on our end and open it back up to all of you. We have a number of modeling opportunities with developing this framework. And we've specifically designed each of these modules and their implementation in the code with extensibility, even replacement in mind. So if you are interested in getting into the code and doing improvements, you should find that doing improvements to the individual modules, or say pulling them out and replacing them with, say, a better idea that you may have, or just simply adding a new mechanism, something like interferon-induced viral resistance, or introducing infection of actual immune cells by the virus, you should hopefully find that adding those new modules and things that may interest you should be relatively straightforward. We certainly invite you to do so. We are very much interested in collaborative development. All of this has been developed with those principles in mind, that we would like to see parallel development. We'd like to see lots of people working on lots of different aspects, and not just on the modeling aspects, but also with identifying experimental data, generating new experimental data. We're certainly interested in developing collaborations and partnerships with experimentalists who either have data available to use with this framework, or who would be interested in generating new experimental data, both to do something like parameter estimation and things like that for calibration of the model, but also to help refine the model and add new missing components and really identify the mechanisms that would actually really define what this virus infection system is. So we're also interested in doing things like cross-platform validation. So we have, of course, our co-authors Lutz and James Osborne. We work with Morpheus and Chaste simulation frameworks. So we like to see these models implemented in multiple simulation environments and using different cell dynamics, both for validation purposes with the implementations, as well as validation of the models themselves. And then lastly, we're also very much interested in using this framework for events like what we've done here today, in more of educational purposes, and not just with how to use the framework itself and how to extend it, but also using the framework as a vehicle to teach how to do computational modeling, how to do abstraction of biological objects and events into mathematical representations. We do a summer course every year for those who are interested in building their own models, really diving into doing the Python-based model specification and development in the CompuCell simulation environment. We do an intensive workshop and hackathon where we start with the basics of Python and we go all the way into individual and team-based hackathon-like model development. I believe this is the 15th year. James, please correct me if I'm wrong. So this year, because of the pandemic, we're doing this virtually. So it'll be a similar setup to what we've done here today, but with pre-recorded sessions and then more personal hackathon-like collaborative development for project-specific code. So you can find more information with our website. Lastly, before we turn it over to all of you to close out, certainly thankful for the funding that we've been provided to do this sort of work, both on the research side as well as on the educational side. And the list grows by the day of the people who we have to thank, who have helped us to develop our understanding of what we've represented in this framework. A lot of friends and collaborators from a lot of different backgrounds, from virologists to experimentalists and other theoreticians and computationalists, so on and so forth. So we're certainly very grateful for all of the contributions that all of these people have made. Certainly not limited to a very brief list that I've included here. So part 2 of the workshop will start next Thursday. We will do 2 different sessions of this part. For Eastern time, we'll do next Thursday at 2 o'clock. And then again next Friday at the same time as today. So this second part, we'll actually dive into the code. We'll see how this model is actually implemented computationally, and then we'll do some exercises on doing simple model development and showing really the basics of how to take an idea of, say, representing a new mechanism and then actually introducing it into the framework that we built. So with all of that being said, we have a few minutes left. James, if you have anything that you would like to close us out with, we have a few minutes for any discussion or last minute questions. Elisa has a great question. Elisa or Liza, I'm sorry if I mispronounced that. Great question. Are we going to repeat the setup of the program in the computer next week? Absolutely. And to elaborate on that more, for anyone who did have issues today, I know it's very frustrating to try to get things set up while you know, everyone sort of moves along. For anyone who did have issues with getting the software working and set up, whether it's a local installation or your Nanohub version, please contact me or Giuliano or Jim Sluka. We're happy to help you get set up and working. We certainly want to see everyone be up and running for when we get into the code next week. You're very welcome. There's my email. So you can pull my email from the chat there. And again, please let me know whatever issues you might have with the software. Yeah, Jim Sluka is also very keen on getting people up and running with the software. So Jim is also happy to help. Yeah, the last thing that we want is for people to show back up next week and then not be able to run anything. So we can get all of those things worked out between now and then. Another great question. The presentation that we went through today, the slides, are they available? They are, and they are available at the Google Drive link where the simulation files were also available. So you can follow the link that I just dropped into the chat and the exact slides that we just talked through are currently available at that Google Drive link. Okay, well, it's 12 o'clock here in Indiana. Unless there's anything else, we'll go ahead and close this out. Thank you again for all of your time and your interest. We hope to see you back here this time next week. And yeah, have a great weekend.